I mentioned Beth. Yeah. We've got night class. Testing, 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 testing. Works. Good, good. All right. Let's go here. There.
Yeah, it's amazing, guys. It's amazing, guys. Let's see. Down.
All right, so question is one. Unboxing A2 is a what? Later. Vasoconstrictor. Today, different things of knitting. Does it feed the dog at night? What is it? Any guesses? Any suggestions? Dilated? Not dilated, no. That one actually could have two answers. Any guesses what they are? They're just fashion glass and all that. You know.
I know, I really am.
Trend. I really wearing one for long.
That's very true.
It's the vaso member. It's made by who makes it? Mast cells make it. Base cells make it. Do red cells make it? Who or what makes unboxing A2? The big hand starts with a P and to the P. Oh, come on, Gavin. Platelets. Platelets make it. The platelets help to staunch bleeding. So remember to staunch bleeding, what does the vessel do? It. There you go. Good job. Very good. Constricts. So this is a vasoconstrictor and it's a platelet activator. So that one or two answers. Either one would work. What's the next one on there? Bradykinin. What does that do? Talk about kinins. What about kinins do? Yeah, there's a dilate, very good. That's one important thing they do. They do one another thing. Anybody remember what that is? Right? Right, Penny. Right, they activate nociceptors. Okay, what's the third one on there? Prostacyclin. Prostacyclin, what's that do? My Bible quizzes with all quotes. Let's see, how to start? It's always the hardest part, how do quotes start? But godliness. Oh yeah, but godliness with contentment is great game. So fine, now what's the reference? Second Timothy? No. Titus? No. Well, it's not Matthew. Yeah, what's left? First Timothy. Okay, which chapter? Prostacyclin. Anybody got anything on that one? What's it do? Endothelial cells, when they're happy, healthy, and whole, they make two molecules. What are they? One of them is made from arginine. Remember we talked about it? Its acronym is NO. Nitric oxide, thank you, nitric oxide. What's the other molecule they make? Prostacyclin. So those keep the smooth muscles surrounding the vessel relaxed. And prevent clotting. And tell the immune system everything's just fine. Nothing to see here. And what's the next one? Thrombin. Thrombin, what's thrombin do?
Would that mean process like orders the thyroid?
Yeah, there's that way. Yeah. Thrombin. Thrombin actually does several things, but what's the most obvious thing thrombin does? Hey, Emma,
Hello.
feeling better?
Uh, it hurts a little more today.
Oh, okay.
Yeah.
Hang in there. When do you start your physical therapy?
I'm setting up my thing, I think, today to get it all set up.
Soon.
Yeah.
Very cool. You're gonna do your PT here with Mitch or?
Yeah.
Excellent. Mitch is really good. You like Mitch. They're doing a self-test. That's why we're chatting. Thrombin, guys. Thrombin, what's it do? Emma, what's thrombin do?
What does what do? Sorry.
Thrombin. 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 Any guesses?
No.
I had that one said. Remember the common pathway, common clotting pathway, common pathway, factor 10. What's factor 10 do? Factor 10 clips prothrombin into thrombin, and then what does thrombin do? It clips. It's 4% of your serum protein in blood. It's called. Sounds like fibrinogen. Very good, Lisa. Thank you. Fibrinogen clips it into fibrin, and the fibrin self-assembles into. Help me out here. An example with the rebar and the concrete. I'm gonna let you guys look at that. Okay, let's do the next one. Complement can be activated. There's 3 ways to activate complement. There's 3 pathways. Remember what they are? I'll give you the first one, the classical pathway, or the other 2. Lectin, mannose binding lectin pathway, good. Third one is the good alternative pathway. Now, what activates each one? Which is which? Classical is activated by who? Antibodies, good, and yeah, C1, greater C1. The mannose lectin binding pathway is activated by the mannose lectin binding protein, which is like C1, but it doesn't bind antibodies, it binds polymers of mannose. And then the alternate pathway is activated by C3, good. Does C3 bind to just any old thing? It binds to surfaces of microbial surfaces. Good. You guys remember that. Pat yourself on the back. Then what else did I ask? Which white blood cells are like mast cells? You wrote basophil. You get the prize, very good. Which white blood cells attack metazoan parasites? Like worms and things like that. Eosinophils, good, eosinophils, very good. Healing requires which growth factors? Just give me 2. Released by platelets. One of the cells that makes the collagen that knits things together is the fibroblast. And one of them is blank growth factor. Fibroblast growth factor, right? And then the second one, because it comes from platelets, it is platelet-derived growth factor. Good, those are two. There are some others, but I take those two ones. New blood vessels are formed by these cells. Anybody remember what they're called? I see a couple of you shaking your head. No, not one of them. We call them EPCs. Remember what EPC stands for? There you go. Very good. Endothelial progenitor cell. Which growth factor activates them to form the blood vessels? We talked about it. VEGF, did I talk about VEGF? No? That maybe that's on me. So VEGF is vascular endothelial growth factor. That's also released by platelets, and that induces angiogenesis, the formation of new blood vessels in the healing process. All right, so that hopefully helped you remember some things from last time. Looking now at our continued discussion of adaptive immunity, we ended talking about active and passive immunity. Active immunity means you made the antibodies, the cells, or whatever yourself. Passive means it was given to you through injection, through intravenous infusion, through the placenta, through the colostrum of your breastfeeding, something like that. That's passive immunity. So if we ask this question, which of the following terms describes a type of immunity that occurs when preformed antibodies are transferred from a donor to a recipient, you would answer, right, passive. Passive. Passive immunity occurs when you have preformed antibodies that are transferred from donor to recipient. And the good example would be the transfer of maternal antibodies across the placenta or something like that. Some terms to get your head around. Antigen. Antigen. A foreign substance that your immune system responds to is called the antigen. An immunogen is an antigen that your body makes antibodies against. Not all antigens elicit the production of antibodies against them. Some antigens just elicit cells that respond to them. If the antigen causes a production of antibodies, then it is an immunogen. So all immunogens are antigens, but not all antigens are immunogens. So immunogens are a subgroup of antigens. Now, another term that your book uses, a piece on the antigen that your immune response recognizes specifically, is called an epitope. Epitope. E-P-I-T-O-P-E, epitope. So if we use, for example, this here so Emma can see. If the water bottle is an antigen, then here's an epitope, the lid here. The handle is another epitope. The central piece is another epitope. And down here, the base would be another epitope. So epitopes are structural parts of the antigen that the immune response will recognize specifically. if your antigen, or if your molecule rather, is too small for the immune system to successfully recognize it, then you will take it and attach it to a bigger molecule. And then the immune system forms an immune response against that bigger molecule, plus the small piece you attach to it. That carrier with the immunogen attached to it is called a hapten. And that process is called haptenization. So for example, you guys, your kids, you were inoculated, you were vaccinated against pneumococcus. You had a pneumococcal vaccine that were capsular antigens, but capsular antigens are carbohydrates that repeat over and over and over and over again. The immune system doesn't do a very good job of recognizing those. So the shot that you were given actually contained a protein to which those capsular antigens were attached. And now your immune system responded against those like gangbusters. Those are called haptens. You take something small or highly repetitive, you attach it to something big and much more interesting, and the immune system responds to the whole thing. So this was explained to me once when I was an undergraduate. I thought this explanation worked really well. So I grew up on a farm, as I told some of you before. And in a packing house, you tend to have little visitors called mice. Mice are really good at being really quiet. And because they're dark and because they tend to hang out in the shadows, they can be very hard to see. And sometimes the mice will get a hold of something that they really wanted, and they start carrying it around. And then all of a sudden, you can see them. And my dad had this BB pistol, and you see them on the rafters sometimes, especially if they're carrying something. He got pretty good at picking them off the rafters. And it was my job to pick them up and throw them away. Great. So the mouse, you couldn't see it, but then it had, we used a lot of boxes because the grapes were packed in boxes. So bits of lumber lying around often. Sometimes they want to make a nest, they pick up a little piece of wood and they carry it around, and that wood, you'd see that piece of wood on the rafter, that's a mouse. Or you'd see it in the shadows, a little piece of wood. Hey, wait, pieces of wood don't walk around, that's a mouse. That's an example of a hapten. So the mouse is hard to see, but you combine the mouse with a piece of wood, and now you see it really well. Not with that. So antibodies are secreted glycoproteins. They're made by B lymphocytes. We won't spend time in this class talking about the complex developmental cycle of B cells. That's for another class. But B cells have antibodies on their surfaces that are receptors for antigens. The antibody genes are quite varied. The B cell actually rearranges those genes so that it makes one specific antibody. And when the antibody is bound relatively tightly by an antigen, it activates the B cell. And the B cell starts to divide, it proliferates, and it starts to take those antibodies which are bound to the surface, and it starts to secrete them. So there are 5 different classes of antibodies, and they all go Ig. The Ig stands for immunoglobulin. Immunoglobulin is another term for antibody, and then a capital letter. IgG, IgA, IgM, IgE, and IgD. Now, when the B cell is very young, it's born in the bone marrow, and it's maturing, rearranges its antibody genes, the first antibodies it will make on its surface are IgM and IgD. And it cannot escape the bone marrow until it makes those 2 antibodies on its surface, IgM and IgD. IgD is never secreted. IgD is just an antibody that licenses the early young B cell to leave the bone marrow and into the circulation. When the B cell is activated with antigen that binds its antibody, it will secrete IgM. IgM is the first antibody it secretes. Therefore, consequently, when you get vaccinated for the first time, the first sets of antibodies that you will make are IgM antibodies. If you draw someone's blood, you find antibodies against something and they're IgM antibodies, that tells you this person has not had this infection before. This is the first time they've had this infection because they're making those early IgM antibodies. As the B cell makes these IgM antibodies, it will rearrange its antibody genes to make either IgG, IgE, or IgA. Now, IgG is usually the one that it makes because that is the most abundant antibody in circulation, IgG. And as it rearranges its antibody genes, it produces antibodies that actually bind the antigen more tightly. So IgG antibodies bind the antigen more tightly than do the IgM antibodies. The IgM antibodies aren't made until later in the infection. So the IgM antibodies will usually peak 8-10 days after the infection, and then the IgG antibodies will start, and after about 2 weeks, they're nice and detectable, and then they will peak, and the IgM antibodies will go down to pretty near nothing. So if you draw someone's blood, you see IgG antibodies against this microorganism, whatever. That tells you they've had this infection before, or they were vaccinated against this infection, or they've had it for a long enough time so that the body is starting to make IgG antibodies. Stop right there, make sure you're with me. Don't pretend this is simple, it's not. Nothing about immunology that's simple.
So the IgD, it secretes the IgM.
The early B cell puts IgM and IgD on its membrane.
Okay.
It inserts it in the membrane. It cannot leave the bone marrow, so that's the passcode,
Yeah.
essentially, for getting out of the bone marrow. So you go to your dorm, you get in your room, you have to have key, and you have to have your ID card to be able to get into the dorm, right? So you need your IgD and you need your IgM, right?
Yeah.
And then the IgM is the one that's secreted. The IgD is never secreted, it's just a licensing tool for the antibody.
Okay.
Now, the B cell might very well, when it rearranges its antibody genes, also make either IgA or IgN. It depends on the type of antigen, and it depends on the signal the B cell is getting from the rest of the immune system. And I'll show you what these antibodies are for, because they're different in structure. So here is IgM. IgM, as you see, is a pentamer. There's 5 different antibodies all linked together. And so this particular antibody is extremely good at aggregation. Extremely good at aggregation. It's also extremely effective at activating complement. So activating complement through the classical pathway. The IgD looks like a typical Y-shaped antibody, except it stays embedded in the membrane. It never gets secreted. And here is IgG. And IgG is an excellent opsonin.
excellent opsonin. And it also is a very good activator of the classical complement pathway. Now, what's going on with IgA here? IgA has this little piece called the J chain. That J chain piece takes these two different IgA antibodies and links them together as a dimer like this. Then there's this other piece here that's a secretory piece. This antibody is secreted across mucosal surfaces. IgA provides secretory immunity.
So what sort of things affect mucosal surfaces? Let's brainstorm here a little bit. Think of the things that make you go achoo. What sort of things make you go achoo? Pepper. You're not going to make very good antibodies against pepper. Molecules are too small. What infectious agents might make you go achoo? Smart aleck. Yeah, colds. Colds are caused by rhinoviruses, coronaviruses, adenoviruses. They're caused by lots of different viruses. So IgA antibodies against the outer coat proteins or glycoproteins in the membranes of those viruses that are enveloped will give you immunity in the upper respiratory tract and prevent the virus from infecting your lungs. IgE antibodies are made against antigens that are found in metazoan parasites. So these will coat the surfaces of worms or amoebas or protozoans or other types of critters that will bump in the intestine or something like that. IgE antibodies are also very important when it comes to allergies. And let me give you a little aside here, because you guys all probably got the COVID-19 vaccination. Yep. A lot of you got it. No? Let's see. A little bit of this, a little bit of this. Well, then let me tell you a little vignette. So, when the reports came out, when the New England Journal of Medicine published the early clinical data on the Pfizer and then later, a little bit later, the Moderna vaccine, it was clear that the vaccines generated a very robust immune response and that the immune response could protect you against getting at least the strains that were present then of SARS-CoV-2. The antibody that the vaccines tended to cause you to produce was IgG. So IgG will do a very good job of preventing systemic infection, but the vaccines did not do a good job of inducing IgA. So you could get a COVID infection, but the vaccine would prevent it from going to the lungs. Because in the nasal cavity, the virus infects the nasal mucosa, divides very robustly, and then works its way down to the lungs. You could still get infected, and because you can still get infected in the mucosal surfaces, you could still sneeze it out and transmit it. And the early papers said nothing about transmission. Certain health officials, who will remain nameless, said that the vaccines prevented transmission, which was bunk. And when that was published, several people Adam Sifu, Bernard Prasad, Jay Bhattacharya, yours truly, said, but nobody cares what I think, said, I really don't think these are probably going to prevent transmission because they don't make secretory antibodies. And that turned out to not be true. And then health policy was made based on this mistaken impression, I'm trying to be charitable, that the vaccines prevented transmission, but that didn't seem reasonable because they didn't induce an IgA response. That's why companies started to look at a nasal vaccine to see if the nasal vaccine would generate IgA, because that would potentially prevent transmission. So there's a little vignette. Reagan, question?
So for IgA, that's the one that if you are producing that antibody, you won't be able to like transmit it.
You'll be less likely to transmit it because the virus will get into your nasal mucosa and it'll be met with IgA antibodies that will prevent it from being able to attach and infect. So any infection you get will be very slight, subclinical, and the amount of transmission would be quite limited as well. But vaccines that are given intramuscularly, it's hard to get them to make IgA antibodies. They usually make great IgG antibodies. That's a tough one. And so folks are looking at using nasal spray, doing nasal types of boosters and things like that, which is pretty ingenious, but quite hasn't worked out yet. So IgG gets transported across the placenta. It's the most abundant class of antibodies, counts for most of the protective activity against infections. There are 4 subclasses of IgG that are just called IgG 1, 2, 3, and 4. They have some slight differences in their capabilities. I'm not going to get too much into those. One thing I do want to say about antibodies, so this piece here is the piece that binds the antigen, this piece here. And so that gets called the antigen binding region, or this fragment here is called the FAB. This fragment from here to here, this is actually constant. And so this particular fragment sometimes gets called the F sub C, C meaning it's constant. Now the F sub C, because it's constant, if antibodies are bound by cells, because there are receptors on cells for antibodies, it will bind this piece here, the F sub C region. So those are called F sub C receptors. And the F sub C receptors help with opsonization. So IgG antibodies, most abundant. IgA antibodies are important in secretions. Name some secretions that might have IgA antibodies, you think? Mucus, very good, Aaron. Mucus, what else? Saliva, thank you. What else? Sorry? Tears, possibly, yes. I don't want to get too racy, but you're going to be nurses, so you might as well get used to it. Semen. Semen might have IgA antibodies. So might cervical secretions as well. So this is responsible for secretory immunity. This type of secretory immunity will also play a role in certain autoimmune diseases like celiac disease. And it's a secretory piece that's bound to it that is responsible for the antibodies being able to be secreted across epithelial layers. IgM is a pentamer stabilized by that J chain. It's secreted early in the life of the B cell. And then IgD, there's a little bit that gets clipped off the surfaces of B cells. So you find a little bit of it in blood, but it doesn't really play a role in immunity. It simply signals the maturation of the B cell. Now, urushiol is the oil found in poison ivy and in poison oak. You don't really have poison oak out here. There's lots of it in California. Believe me, I was well acquainted with it several times. And this is an example of an antigen that does not produce an immune reaction. Is that true or is that false?
This is actually true because not all antigens are immunogens. Urushiol does what it does, causes, if you get poison ivy on you, you itch, you get the welts and stuff, because urushiol reacts with cellular proteins and derivatizes them, and that's what your immune system responds to. And as we'll see, that's called a delayed type hypersensitivity reaction. And if you burn poison ivy, you can actually breathe this in and get a very severe respiratory reaction as well. That's why you shouldn't burn poison ivy or poison oak for that matter. So the back part of the antibody that binds the cells, that's the F sub C region. The part up front that binds the antibody, that is called FAB. And so antibodies have two heavy chains, two light chains. And you have both heavy and light chain genes for antibodies, and both of those get rearranged in the B cell. Both the heavy and the light chains have regions that are constant. They're constant in all the antibodies. So every IgG1 at least, the amino acid sequence of its heavy chain, they're all the same. The part that varies is the part at the front, that's the variable region. And the variable region has bits and pieces that vary tremendously because of the gene rearrangement, and those regions have a shape. That shape will fit the antigen. And so you can see here, here are your constant regions here. And then this is your variable region up here. And that variable region, there's lots of different genes for the variable region that get put in and then get rearranged to be associated with certain heavy chains and things like that. Notice that they have a particular molecular shape that fits the antigen quite nicely. So we talked about epitopes already. The epitope is that piece on the antigen that the immune system recognizes. The paratope is the shape that fits the epitope on the antibody, and the antibody more or less binds it via some sort of lock and key mechanism. The association between the antibody and the antigen are non-covalent interactions. So they're hydrophobic interactions, van der Waals forces, ionic interactions, ionic bonds, things like that. Antibodies will bind to things and neutralize them. They will clump things together. Agglutination, when you clump things together, you make them more easily recognized and disposed of. And they're used for precipitation. Precipitation is used more in the laboratory than in your body. But they're really effective for that in the laboratory. Indirectly, they activate complement. They opsonize things, so they facilitate phagocytosis between antigens and phagocytic cells. And they can be mediators of inflammation from time to time. Reason they can be mediators of inflammation is that if they bind to a target, that target gets destroyed. Now these molecules are strewn about. Those molecules that are strewn about can initiate the inflammatory response. So they indirectly initiate, promulgate, maintain inflammation. And this is a picture stolen shamelessly from your book that illustrates that. So they can bind to the surfaces of bacteria, they can bind to the surfaces of viruses, they can bind to the surfaces of worms, they can bind to toxins made by bacteria and inactivate them. They can do a lot of really cool things. And IgE defends against parasites, but it's also involved with allergies. And IgE molecules bind to the surfaces of mast cells. Mast cells have an antibody receptor specifically for IgE antibodies. When those IgE antibodies are bound on the surfaces of mast cells, the mast cell will degranulate. And that mast cell degranulation releases histamine, and the histamine can cause bronchoconstriction. That's the basis of asthma and various types of seasonal allergies. Mast cell degranulation in the nasal mucosa causes the nasal mucosa to run. Mast cell degranulation in your eyes will cause the vessels in your eyes to enlarge, and then your eyes start to tear, and you get exudate from your eyes, and then you get more mucus secreted from the goblet cells and those are seasonal allergies. Some of you probably have seasonal allergies, and you're miserable. And what do? You take an antihistamine that blocks the H1 receptors so that you don't get that response. And IgE antibodies also will help attract the eosinophils, and they will help strengthen and augment the activity of eosinophils against metazoan parasites. So in the mucosal system, you have lymph nodes. They form a series throughout the mucosal system. This is called the MALT system, the mucosal associated lymphoid tissue. Mucosal associated lymphoid tissue. Lymph nodes, and as part of the MALT system, are filled with B cells that secrete immunoglobulin A. And you can see the MALT system is found throughout the mucosal system. You also have a series of lymph nodes that are found in the GI tract. You have lots of them in the small intestine, but they're more concentrated in the large intestine. And this is the gastric-associated lymphoid tissue, or the GALT system. And the GALT system will contain B cells that will secrete IgA and also IgG antibodies. And in the small intestine, you have these small sections called Peyer's patches. And the Peyer's patches have something called an M cell. And the M cell this M cell is like a macrophage, and it sticks up with a very ruffled membrane, and it's going to sample antigens that are found in the GI tract. And these Peyer's patches are important mediators of food allergies in many cases. But they're also trying to detect if you have pathogens in your GI tract. They're part of the GALT system, gastric-associated lymphoid tissue. So when lymphocytes are activated by antigen, and you must remember the immune response is antigen driven. It's antigen driven. You supply the immune response, immune system with antigen, you get an immune response. As everything gets killed off, you don't want the immune system to just keep going. It's a waste of resources. When it's clear from your body, the immune response now will decrease. So it is antigen driven. As the cells are activated, they will divide. So the B cell that makes that antibody, that binds that antigen, it divides and it undergoes so-called clonal expansion. All the cells that are made as it divides
make the same type of antibody that progenitor B cell made, clonal expansion. Now, here's the hitch about B cells. As those cells divide and they replicate their DNA, the DNA polymerase that makes their antibody genes is a special DNA polymerase that makes more mistakes than the usual DNA polymerase. So the antibodies that come out will tend to differ a bit from the progenitor B cells. Some of them will bind the antigen poorly, and if that's the case, those particular cells won't be activated anymore. They'll just go off somewhere and die. The other cells, however, will make antibodies that bind the antigen better. The better you bind the antigen, the more you're activated, the more of your progenitors that you make. And this is called clonal diversity. So with that clonal expansion, you also get clonal diversity because the DNA polymerase that replicates the antibody genes is more error prone and it makes mistakes. Remember that both B and T cells are born in the bone marrow. They're born in the bone marrow. There is a hematopoietic stem cell that makes all your blood cells in the bone marrow. And when that stem cell divides, it will either, depending on the signals it gets, make members of the myeloid lineage or members of the lymphoid lineage. And the B and T cells and the natural killer cells come from the lymphoid lineage. So the B cells, they hang out in the bone marrow and then they attach to a stromal cell and they rearrange their genes and then they figure out their antibodies and what have you. Once they make IgM and IgD, they hightail it out of the bone marrow to the peripheral circulation. The T cells leave the bone marrow and go to the thymus. That's why they're called T cells. T is for thymus. Thymus is a gland right over your heart. Yours is still active and ticking. Mine has probably involuted down to nothing. Think of the thymus as T cell university. T cells go to the thymus to get educated. And so the thymus is one of the so-called primary lymphoid organs with the bone marrow. Because the bone marrow will birth the cells, but T cell university is the thymus. All right, so we talked about clonal diversity. Central tolerance refers to a phenomenon whereby when the B cell is developing in the bone marrow, as it makes its antibody, if that antibody recognizes any self-antigens in the bone marrow moves in quickly and kills it. It will not permit cells that recognize self-antigens. Likewise in the thymus. As the T cell gets to the thymus, starts to figure out its T cell receptor. If that T cell receptor recognizes any self-antigens, the thymus comes in and gives it the deep six. And in fact, the majority of the thymocytes that go to the thymus, the majority of them get eliminated. That is central tolerance. Central tolerance. Immune system. That there was an old commercial with Orson Welles. It was a wine commercial, and he talked about how this company will not let any wine be sold before its time. The wine has to be properly aged and proper passed in order to have the phenolics in it to give it proper aroma and bouquet and taste and yada, 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 yada. Well, the immune system will let no cell go before its time. It needs to be properly vetted. Have to have a proper receptor. The receptor has to not interact with self-antigens. Only then can it join the elite groups of lymphocytes. Okay, that was corny, but it makes the point. So yeah, Aaron. Autoimmune diseases tend to be associated with particular genetic conditions, genotypes, certain genotypes. We'll get to those in just a bit. And certain genotypes tend to allow more release of autoactive lymphocytes. So the T cell, as we said, goes to the thymus, and it also has an antigen receptor called the T cell receptor. The T cell receptor is never secreted. Never secreted. Hang in there, Kylie. I know. A lot of stuff. I feel that way too. I really do. Do you have your only class, Kylie? No? Why are you yawning? And in the thymus, it rearranges its genes to make the T cell receptor. And only when it makes the proper T cell receptor complexed with certain proteins will it be able to leave the thymus and go to the peripheral circulation. Now, little thing about the T cell receptor. The T cell receptor is going to be complex with one of two proteins, not both. Not both. These proteins are CD4 and CD8. C stands for cluster of differentiation. That's just an old name given to it. Don't mind that. It's just a name for a cell surface protein. If the T cell receptor is complex with CD4, that cell becomes a T helper cell. If that T cell receptor is complex to the protein CD8, that T cell becomes a T cytotoxic cell. And all this gets decided in the thymus. So when you're in school, you get asked, what do you want to be when you grow up? That's kind of what the thymus does to the T cell. What do you want to be when you grow up? Well, I want to be a T helper cytotoxic cell. The thymus says, nope, have to be one or the other, can't be both. And if you are both, you're not going to leave here alive. So the T cell has to decide. So the initial, the so-called primary immune response is generated by the initial exposure to the antigen. There is a lag period. This is the time during which B cells have recognized the antigen and they are starting to divide. So about a week or so after you're first exposed to the antigen, you have IgM antibodies that are made. And the IgG response will follow usually about a week later. This is the reason why you're usually told you need about 2 weeks after vaccination before you have good protection against that infection. Here's the reason why. Takes a week for IgM, takes another week for the IgG to come on board. Now, after the initial immune response, the primary response dies down, most of those B cells will actually become quiescent and will undergo cell death. A fraction of them, however, will become quiescent and will become memory cells. Now remember, these cells don't make IgM that fits the antigen disorder well. These cells make IgG antibody that fits the antigen extremely well. So these memory cells can make antibodies like that, that fit the antigen extremely well and provide very good immunity against it. Consequently, your secondary immune response is faster and is more efficient than your primary immune response. And gang, this is the whole strategy behind vaccination.
So memory cells, you have them floating around your blood right now. And as nursing students, when you're after your second year, you get to go into the clinic. But before you go into the clinic, you have to have all your shots, and you're going to get these rather frantic emails from Mindy saying, Zoe, your Hep A is not up to date. Get your Hep A booster. Now, Zoe, today, Reagan, your Hep B is not up to date. Aaron, when was your last tetanus shot? And you'll get these emails from her because you have to have those before you can walk in the clinic. Reason you get them is with that shot, you get this lickety-split immune response that gives you very good protection. Now, when a B cell interacts with an antigen, the B cell may typically react to that antigen, but T cells have to have antigen presented to them. So the antigen has to be phagocytosed and processed by antigen presenting cells. And the antigen presenting cell that will usually present it to the T cell is a dendritic cell. Talked briefly about those last time. Dendritic cell. Here's what antigen processing consists of.
You take the antigen, you chop it into smaller pieces, and you stick it on your membrane. That's antigen processing. Now the T cell comes and it sees the antigen. How does it see the antigen? It sees it with its T cell receptor. If the T cell receptor fits the antigen, T cell is activated. It starts to divide and proliferate. And through proliferation, the T cell now, you make lots of those T cells. And those T helper cells will secrete cytokines that will activate B cell maturation and proliferation. So the T helper cells help the B cells do their job. They also help other white blood cells do their job. They great helpers. T helper cells would make great nurses.
When a B cell differentiates into an antibody secreting cell, it's known as a plasma cell. A plasma cell. Now, what about these T cytotoxic cells? What are those guys about? Here's what they're about. T cytotoxic cells look for virally infected cells because virally infected cells tend to take their viral proteins and put them into the membrane of the infected cell. And if the T cytotoxic cell has the antigen presented to it and it becomes activated, and it sees those viral proteins on the surface of the cell, the T cytotoxic cell destroys that virally infected cell, thus causing an abortive viral infection. There's another group of cells that express weirdo proteins on their surfaces. Cancer cells. And cytotoxic cells are in a constant mode of surveillance. Or cells that
oddball proteins on their surface. If it finds them, it destroys them. The other cells that do that are those natural killer cells. Natural killer cells are just kind of surveying. You don't look right. You're damaged. Let me ease your pain. It secretes poisons that kills the cell. You don't look right. Oh, but I feel fine. You're a cancer cell, and it secretes poison that kills them. So this immune surveillance is extremely important in preventing cancer. This is why cancer tends to be a disease of the aging. Because as you get older, your immune system gets weaker. And immune surveillance tends to really slow down. Now, major histocompatibility proteins. I'll just have time to start this. Won't be able to finish this, Aaron. This actually gets to the question you asked. What would happen if, say, Zoe was in a car accident, she was badly burned, and she needed a skin graft, and Lisa Levine, her dear friend, decided to volunteer some of her skin. She did, I got her skin, and she did have some. Now, if we grafted some skin from Lisa onto you, I mean, that's so kind, but what would it take? There's a really good chance it would. The reason for that is that the immune system, one of the things it reads when it surveys your body, is just looking at these barcodes on the surface of the cell. And those barcodes are these major histocompatibility complex D bodies. And they are referred to as HLA proteins. HLA proteins are encoded by the MHC genes on chromosome 6. There's two types of HLA proteins, class 1 and class 2 proteins. And they're really important in antigen presentation and in determining self from non-self. That's where we will leave this for today. We'll pick this up on Friday. Don't forget, got a quiz on Friday, so come ready. Same quiz as last time. And these little self-quiz questions I give you, those might be, you might find one of those on the quiz. Emma, I will email you the quiz like I did last time.
Okay, perfect. Thank you.
And that's it for today. So. Stop.
All right. Thank you again for Zooming.
Yeah, no problem, Emma. And if you have any questions, feel free to email me questions, okay?
Alrighty. Have a good day.
Okay, everyone say bye to Emma. Get well.
Thank you.
Heal thyself.